Hello friends, welcome back to Smriti Notes for Biotechnology. In today's lecture, we will study homoplasy concept, identical markers versus non-identical markers, and gene annotations. So let's start with homoplasy. The character shared by a set of species but not present in their common ancestors, like the evolution of an eye. There are two main types of homoplasy, that is convergence and reversion. Convergence means that the evolution of same character in at least two distinct lineages. Reversion, that return to an ancestral character state. These topics are not very important, but as it is mentioned in Amity syllabus, so I've mentioned about them from books and some useful links, so you can just go through them, because usually we students find difficulty in finding information about these topics. Next is identical by state versus identical by descent markers. Identical by state means two individuals sharing an identical single nucleotide polymorphism position are identical by state for that SNP position, since no evolutionary information can be inferred. They are used in genetics to describe two identical alleles, and that's why they are called as identical by state when two or more individuals have identical nucleotide sequence. The next is identical by descent. Retrotransposons by insertions are identical by descent markers. The random chance of two individuals sharing same retrotransposon insertions are exactly the same position is almost zero. It is used in identical by descent segment detection is to quantify relatedness used in forensics genetics and IBD mapping. So we have learned the difference about both identical by state versus identical by descent. This much information is more than sufficient. Next topic is microarray based SNP technique. SNP single nucleotide polymorphism is basically a DNA microarray which is used to detect polymorphism within a population. It involves various techniques like DNA hybridization, fluorescence microscopy, and solid surface DNA capture. It has three major components, that is the immobilized allele, the fluorescent dye, and the detection system that records and interprets the hybridization signal. The next topic is gene annotation. Annotation means adding specific structural or functional information about a specific region of that genome. A complete genome sequence by itself is just raw data. So the functional gene involved in metabolism and molecular function. And to identify the gene location and function in the genome, we use gene annotation. These are some that type of concepts that are difficult to find on the internet as well as on books. So I've used various sources and collected the information and written them. You can use this, and if any question comes, you can write all the data. And to get updates of more such videos, do press the bell icon and do not forget to like, share, and subscribe.